We were all very unhappy when Josep Penninger left Toronto, left the Amgen Institute. He had been seduced by his native Austria to come back to Vienna and launch one of the great scientific institutes, an Institute of Molecular Biotechnology in Vienna. Just a huge budget, untold opportunity, and so he went. But he's a cardiac Canadian now, a cardiac Torontonian in his heart. And so he maintains a position at the University of Toronto and comes back several times a year. He's been on this stage at least 2 times previously. 2, yes. The first time he told us about the fact that he had isolated the gene for osteoporosis. This was really a significant scientific breakthrough. We all know the devastation that that disease creates among women, and Joseph's insight really provides the basis for a cure. The second time he came along, he told us about the fact that he had isolated the gene for death. And we will talk about this in the future, but there is a growing body of opinion that death is not preordained. It's a disease like any other, and it might well be overcome at some point. So now Joseph is with us again, courtesy of the Austrian consulate. We've had a little support from them, and he has still a new discovery, an amazingly productive guy, Joe Penninger.
So that's what I do. And actually, since I won the telescope, I might become an astronomer after all. And maybe looking up to the skies, I might see Moses in the Taurus. So I'm really nervous to be here. So this is what interests me since many years. I want to figure out how genes work and how diseases develop. And as Moses said some years ago, I actually went back to Europe to start a new institute. So 5 years ago, we had nothing. We're actually now the largest Howard Hughes-like institute in our country. We built the first lab for kids. We had more than 10,000 kids already in our place where we teach them science and genetics. And the philosophy was very simple. Hire the best people, give them enough money, and let them loose. And this is what we want to do. So we want to make a functional map of the genetic world. Oh my, I'm really nervous. So you all know, we know now genes for humans, for mice, for bacteria. When Bill Clinton announced the Human Genome Project, it took more than 13 years to finish $2 billion. It was thousands of scientists who did this. Today, we can actually sequence a human within a week for around $10,000. So we're in the middle of the genetic revolution, and it basically allows us to scan entire genomes of organisms, and recently actually learned something really interesting. So if you would actually take me, grind me up, and sequence what comes out at the bottom, you would actually only find 1% human DNA. And this really keeps me humble, because in essence, the other 99% are bacteria and viruses which live with us. And I think Douglas Adams probably had it right. He just got the wrong species. So it's bacteria and viruses which run us as humans. And you know, when we die, they find somebody else. They let us move around so we can eat for them. They might have even invented sex so they can visit the neighbors. And maybe Viagra to visit them a little longer. So that's what we do. And actually the technology to figure out how genes work, because we're sitting on this millions of information And actually, if you would take all the DNA in my body, you could travel 300 times to the moon and back. So this is what we all have in our DNAs. And actually, the way we try to find out how these genes work, because that's the real question, what does it all mean, is this technology. We make knockout mice, so we take a little embryo, take stem cells, so we learned this very early on. We had, of course, no idea that this would lead to cloning and other things. You can change these genes in little stem cells and literally take out any gene you like, put it back in the early embryo, and make a new organism. And the organisms we are creating is a little mouse. And this is a mouse we made some years ago when I was still in Toronto. And actually, one day we made these little guys here, and we hit a gene which made the mice very tiny. The heads had changed, they actually didn't have any teeth anymore. And the reason was this one: we hit the master gene for bone loss. And when we published this, the media basically said we had identified the osteoporosis gene, and it was actually really nice, because it's a gene which then makes and codes for a protein which tells our stem cell to become a bone-eating cell, a cell which is called an osteoclast. And there are around 300 million women out there which have osteoporosis. People with arthritis are crippled, kids with leukemia are tiny and small most of the times because of the leukemia. And so we basically had found this in the mice. And the other nice thing was it basically works like a funnel. You can throw in and blend in 100 reasons for bone loss, and all of them have to go through the same gene. So we basically hit the mother lode. And there were some other interesting issues. Because when we found this, we actually realized that the same gene and the protein was made in our white blood cells. And at that time, we asked a very, very heretic question. So could it be that our white blood cells actively control how much bone we have in our bodies? And it indeed worked. So I can do the experiment, and each of you take a little piece of your blood, there are actually stem cells in there, put these white blood cells on with this little protein on them, and they would turn into this beautiful bone-eating osteoclast. So we can go to the butcher, buy a bone, and you can actually watch them, how they eat the bone away. So why would this be actually interesting? This is rheumatoid arthritis. It's around 1 in 100 people who get it. It starts with inflammation, so white blood cells basically going into the joints lead to pain, lead to destruction at the end of the day of the joints. And so we thought, if this protein, which we call rank ligand, is the essential molecule which triggers bone loss, and white blood cells make it to turn on the bone-eating cells, so this could be the principle how inflammation, rheumatoid arthritis, actually leads to crippling. So we did the experiment, and indeed worked. We could completely shut down crippling in rheumatoid arthritis. And so based on this, we actually believe we have a rational explanation for osteoporosis. Every third woman, aged woman, will actually get bone breaks because of bone loss. So the genetic principle is the molecule we found here. We have a molecular and rational explanation why inflammation leads to crippling and arthritis. We can actually predict who will lose their teeth and who will not lose their teeth based on this little protein. We know now that children with leukemia are mostly tiny, have brittle bones, because the leukemia cells making this protein, which then turns on the bone-eating cells. HIV patients, AIDS patients, they survive no longer. They have a real problem now. They have bone loss. Turns out the virus turns on this protein, which leads to bone loss. We heard about space flight and the problem of osteoporosis and bone loss. So half a year in a space station leads to around 6 to 7% of bone loss. So that's a real problem because people could not walk anymore. Turns out when we shut this down, the system, you can completely shut down bone loss in space travel. So we can indeed populate space in the future. Material scientists have now started to work on implants, tooth implants, hip implants, because it turns out different materials turn on this protein, which explains why certain materials lead to bad hips and replacements and others too. And the reason why I'm telling you this, a big American company picked this up, made a medicine based on the principle, so a rational medicine to basically completely shut down bone loss in all of these diseases. And they already went through phase 3 clinical trials, and the data, the 2 injections a year, might be sufficient to wipe out osteoporosis. So they applied for FDA approval, so it's FDA, but it might come on the market already this year. So the question was, so we heard about building a robot. So if you can build a human robot and make basically a skeleton, so why would you put a skeleton which allows us to move under the control of sex hormones? So what would be the evolutionary advantage or disadvantage of doing this? Because if you build a robot, you probably would never even consider this. And so it turns out the same molecule which controls how much bone we have is absolutely essential to form a lactating mammary gland in mammals. Basically, the sex hormones in pregnant women turn on this protein in the mammary glands. So the mammary gland cells start to grow, produce milk, and that's the only place where the babies get the calcium from. So basically, evolution has taken the same molecule to release calcium from the bone of the women when they're pregnant and when they lactate, and uses the same molecule to form the tissue, to pass on calcium to the kids. And of course, the kids need calcium so they can walk and are healthy. So we believe that's the molecular explanation why bone loss is under the control of sex hormones, and why older women in particular, the men also get bone loss, but at that time we already have died of prostate cancer. So why this is got under the control of hormones. So this took us straight into breast cancer, because basically what we had seen was that this molecule responds to sex hormones and drives the growth of mammary glands. And there's a lot of data out there that because of hormonal replacement therapy in older women, the incidence of breast cancers have increased. And I talked recently with a researcher in Australia, and he told me in some countries, hormonal replacement is actually reduced now, and the incidence of breast cancer has gone down by 15 to 20%. So it's very clear that hormones drive breast cancer. So it turns out you can actually drive breast cancer in the little mice I showed you, and you normally get 100% incidence of cancer if you give them a hormone called progesterone and something nasty on top of it. And when you shut down the system I showed you, breast cancer incidence went down from 100 to 5%. So this might be the principle how hormones couple to drive the breast cancer cell into the cell cycle and to transformation. So people are actually actively thinking one could run cancer prevention trials based on that principle. So one thing we noted and published was this one, an old idea of Stephen Paget. So he found more than 120 years ago that certain tumors love to metastasize into certain tissues. So women with breast cancers get metastasis into the bones, men with prostate tumors get metastasis into the bones. And so we thought this looks interesting, because rank ligand, this molecule I showed you, controls the bone environment. It controls also how breast cancer cells and breast cells are growing. So this could be a soil factor, how tumor cells feel comfortable to go into the bones. And if we switch it off, we might be able to selectively interfere with metastasis into the bones. So we did the experiment, and it actually worked. So now there are large phase 3 clinical trials going on with cancer patients who have no metastasis yet. In these patients, the system is being switched off. And the hope, of course, is that all our data we have now generated in animals can be directly translated into humans. And of course, the beauty is we have indeed a medicine. So this is not just a little pitch of a future development, but this probably is real later this year. So this is what interests me now. There are a billion people who are obese, and we got into this because we wanted to address this question. So how does a Homo sapiens turn into Homo sapiens? And there was this idea coming out of Harvard that changes in energy-producing mitochondria would lead to this. So we actually, for the first time, modeled it and could show we found exactly the opposite. But what was interesting, when we did this and hit the same genes in fruit flies, in Drosophila, we could actually make flies which were skinny or fat. So we thought this is really interesting because this might allow us to use insects to model human obesity. So we hired a guy in Vienna, and he made for us the first full mutant fly library in the world. So these guys made actually 20,000 individual fly lines where we can switch off every gene we like in any tissue we like, and basically allows us to do entire genome mining for certain diseases. So we did this for obesity. This is what you get. So we have hundreds of genes which control obesity in flies when you shut them down in the brain, in the fat tissue, in the liver, or in the muscle. And actually one of the genes we then took, went into mouse stem cells, made a knockout mouse, and generated a new animals, and basically asked the question, can you directly translate data from Drosophila, fruit flies, into mammalian biology? And I should also say that 70% of fly genes you can actually find again in humans. So you can give flies diseases, put back the human gene, and they become normal again. And so this is what we got. We got the mouse which was entirely skinny. So here we switched off this gene, and actually was really cool. We have two types of fat cells in our body. So there's these white fat cells which make us fat, and there's a good brown fat cells which actually burning a lot of energy. And so it turned out we actually generated a mouse which basically didn't have any of the bad fat anymore, but the good brown fat was entirely normal. So the fruit flies basically might have shown us a pathway in the future how with some little fiddling and drug development we could actually fool the cells to become cells which burning fuels instead of getting cells which are storing fuels and make people obese. Another thing we modeled recently was actually heart failure. So what you're seeing is a fly heart pumping. So this is actually how our hearts looked when we were very tiny embryos. Only later our hearts started to twist and turn a little bit. So the fly hearts, the beats are very similar. And so we also did entire genome mining for heart failure genes. And so this is actually a fly we made which has heart failure. So the one on the bottom, you see the hearts are much bigger, the pumping irregularly. So again, we believe you can actually model diseases and mine entire genomes to model heart failure. We did it for obesity. We developed a fly model for diabetes. We developed a fly model for cancer metastasis. And we also let the fly model smoke. So we are not... It actually blows the brains right out. And the reason is because nicotine is a natural insecticide. So what plants make to fight insects off. So, and my last little thing is we also made a genome map for pain perception. So something which interests me. So most people have to live with chronic pains. And so we thought, let's develop a fly model for pain perception. And so we did this, and this is actually the fly brain. And these little colorful dots are all the brain regions in the brain of the fly which control pain perception. And 2 million experiments later, you get the Jackson Pollock painting. And basically, it allows you to create an entire genome map of pain perception. And of course, the next question is, can you take this again into humans and animals? So one of the genes we found was this one, a gene which was called straitjacket, It's again the fly brain, and these dots are the regions in the brain where this gene straitjacket makes a protein, and these are the brain regions where straitjacket control pain perception in the fly. So we actually took this gene, took a mouse embryonic stem cell, shut down straitjacket, made a knockout mouse for straitjacket, and we got a mouse which had exactly the same what we saw in the flies. So they had reduced perception of pain. So the system indeed works. But there was one thing which we could not figure out well. So straitjacket was only in the brain. So we needed to model actually pain perception in the central nervous system. So we moved our animals in a functional MRI machine and made a 3D reconstruction of all the brain regions which are turned on if the animal feels pain. So this is a map, a 3D map. It's actually not published yet. Nobody's ever done this before, of pain perception in the head of a mammalian species. And the real interesting surprise was when we looked in our mutant mice and we gave them pain, and actually turns out instead of sensing pain, when we shut down this gene called straitjacket, our mice started to hear, smell, and see the pain signal. So their brains got completely rewired, and there's a sensory stimulus like pain, which is essential for us. So I need to know this is hot, because otherwise I'm in trouble. I actually got rewired. And instead of being sent to the right regions in the brain which process pain, so I can move my hand away, I can feel it, these guys started to smell, hear, and see it. So this is a condition called synesthesia. If you look in the literature, there's not a single animal model ever published on this. It's a condition which is known for the 2,000 years. Famous composers like Messiaen had synesthesia, so he said he could actually see his symphonies. So the people out there actually read something and can hear a certain word. So the people out there who have a pain sensation and they can hear it. And I also wanted to show this because I think ideacity is something like this, where we're exposed to different sensory inputs. And I think good meetings work because they actually channel the stimuli into other places. And synesthesia has always been associated with creativity, because, of course, if you start to see notes, or if you start to actually smell, if you have a pain signal, then your brain might be a little wired differently from other things. So this was my story. I believe modern genetics is you said before, where our kids should be. I think this is where our kids should go. Biotechnology has an incredible promise to make new biofuels. The little thing I showed with the drug development for this osteoporosis gene we found, the analysts already estimating around 2 billion of revenue if this comes on the market. So with biotech and genetics, one can not only solve issues which we could not even dream about some years ago as medicines for humans. So if this indeed works and comes out on the market, in essence, we might be able to improve lives of hundreds of millions of people. Old women who actually able to walk, who don't have breaks of the bones anymore. And of course, what's also nice, you can actually make a little money with it. And maybe my final thought, because give you some little idea, and it's actually not my idea. It's from one of my favorite books, the Book of Restlessness of Fernando Pessoa. And when I look at this meeting, which was absolutely tremendous, with a lot of ideas, with lots of brilliant people who were speaking, and of course with all the challenges we have been facing and we are facing. So Fernando Pessoa actually wrote in his book that we all live on this boat in the middle of the ocean. We have no idea where we come from, and we have no idea where we're going to. So the least we can do is to be a little nice to each other.
Is that your soccer club?
Yeah.
Yeah?
Yeah, this is actually...
Obama shoots hoops and you play soccer with the president of Austria, don't you?
Absolutely, yeah. And that's very important for me. It's the shirt of our soccer team, the last time we won against Germany,
Right.
so, which was 31 years ago.
Thanks, Joe.